I'm really excited to introduce Dave O'Connor, whose group on the UW-Madison campus has been working with the CHTC, HTCondor, and high throughput computing over the years for their computational work relevant to a variety of viruses in human and non-human primates. Dave's work is well respected across the globe, including contributions to the scientific community's understanding of viruses like HIV, Zika, and COVID-19. In the last year, Dave's group used more than 165 million core hours, mostly for sequencing the COVID-19 virus, contributing both to local COVID-19 pandemic response and to the greater understanding of variant spread more broadly. I'll let Dave introduce his numerous affiliations and partnerships as he needs to at and beyond the University of Wisconsin, but we're very excited to have him as this year's plenary speaker to kick off HTCondor Week, and for all of you to get to hear about the impact of high throughput computing on his important work. Take it away, Dave.
Thanks, Lauren. Can you hear me okay?
Yes.
Okay, I'm gonna try and see if I can just share some pictures here. Can you see this okay now?
Yes.
All right, well, thank you everyone, and especially to the organizers for giving me the opportunity to tell you a little bit about what we've been doing over the last year or so. As Lauren said, this has been a huge big tent project that's involved not only my lab, which is shown here, who have had to scramble to do work under really challenging conditions, but also my colleagues at the AIDS Vaccine Research Lab, Tom Friedrich and Shelby O'Connor, our colleagues in Public Health Madison-Dane County, as well as the City of Milwaukee Public Health Lab, the Wisconsin Veterinary Diagnostics Lab, Wisconsin State Lab of Hygiene, Department of Health Services, and many others. In terms of disclosures, we received a Promega Maximo instrument as a gift in kind from Promega at the start of the epidemic, and I am a honorary professor at the University of Melbourne. None of those disclosures have any bearing on what I'm going to talk about today, which is really our work around genomic surveillance of COVID-19. And so it's really wild for me personally. I've been sequencing viruses since I started grad school in the late 1990s, and I spent the first 15 years of my career sequencing HIV and SIV, its monkey cousin. And most of that time was spent toiling around in relative anonymity. Certainly things like viral sequencing weren't front page news, and people weren't breathlessly following the results of viral sequences and viral variants as they made their way around the world. And so it's been quite a journey in terms of how we got to this point from where we started. And so it was fairly humble beginnings. Not sure if you can see the part on the right here, but this is now Dr. Gage Moreno. He successfully defended his PhD last Wednesday. He was a grad student in the lab who had been doing viral work and offered to help start sequencing SARS-CoV-2 early in the pandemic, not knowing quite what he was getting himself into. And as you can see here, in the middle of February of last year, we got our first sample into the lab where we were asked to sequence it and figure out what the genetic code is at each of the 30,000 letters that comprise the SARS-CoV-2 genome. And fortunately, right around this time, a group based out of the UK had developed a protocol for sequencing COVID-19 genomes. And the basic premise of the protocol is that you get a nasal swab or you get some other sample, you extract the virus's genetic material, you perform an amplification reaction that amplifies an itsy bitsy part of the virus, about 400 genetic letters. And you do about 120 of these PCR reactions or amplification reactions in parallel. And that together covers the 30,000 base pairs of the virus's genome. You then prepare what's called the sequencing library, which is adding chemicals and reagents so that you have the ability to read out these letters on DNA sequencers. And fortunately, the arrival of the pandemic coincided with the recent technological advances in what's known as deep sequencing that allows you to generate large amounts of DNA sequencing data very quickly. And in particular, one technology, which is known as the Oxford Nanopore technology, allows you to collect the data truly in real time. And what I mean by real time is as the DNA is sequenced on the instrument, every 4,000 data points that come through generate a single file that has the A's, C's, T's, and G's for those 4,000 sequences, and you can analyze them as soon as they come off. So if you start the instrument, within a couple of minutes, you'll have the first file with the first 4,000 sequences, and then you'll have the second file with the next 4,000 sequences, and so on and so forth, which really takes a process that used to require an instrument running for a week or longer before you got data off of an instrument run, and it reduces it down to a matter of minutes until you start getting data off these machines. But at that point, you still have to do a fair amount of analysis, and this is where CHTC has been absolutely vital in our work. So after we get these sequences, we have developed workflows on CHTC that quality control the sequences, that take the sequences that are in a particular instrument run, which we feel kind of like a school bus with a bunch of different samples that are can be differentiated from one another through the use of molecular barcodes. So individual sequence tags that say, okay, well, if this specific tag is seen, that means it came from sample 1. And if this specific sequence tag is seen, it came from sample 2, and so on. So we need to demultiplex those barcodes. And then we need to determine which part of the viral genome a sequence is aligned to. So we map the reads to a reference genome. And then we need to say, right, so now we have all of these sequence reads from a particular sample. How do they, what sort of genome does that make? What sort of variants differentiate this particular genome from this particular individual from other genomes that have been sequenced globally or in our community? So SARS-CoV-2 is now the most widely sequenced virus in the world. There's more than 1 million such sequences in one of the public repositories. And so lots of labs and lots of different places around the world have been generating these sequences. And so we figure out what the sequences are in each of our samples. And then we do a bunch of comparisons asking how a particular individual's virus sequence fits into the broader context of what's going on elsewhere in the world. And so to date, we here in Dane County, Wisconsin, for those of you who don't know, Dane County is the county where the University of Wisconsin-Madison is located. We're a county of about 500,000 people. And since the start of the epidemic, viral sequencing of this sort was really decentralized in the United States. There wasn't a whole lot of coordination between different groups that were doing this. You had academic centers that had previous viral sequencing experience with other viruses, like my experience with HIV and Zika. My colleague Tom Friedrich had sequenced a lot of influenza. And so you had very uneven coverage of virus sequencing. And so early in the pandemic, the lessons that we learned were really important in understanding what was going on in our community. While there wasn't that much data coming in from other similar communities around the country. So to put it in perspective, throughout 2020, we were sequencing somewhere between 3 and 5% of all of the COVID test positive cases here in our county, whereas nationwide, less than about 0.1% of all cases were being sequenced. As we moved into 2021, there was a real resurgent interest in getting more of this sequence. It was catalyzed by the discovery of these so-called viral variants or variants of concern. And now nationwide, we're at about 3 to 5% of all test positive cases throughout the entire country are being sequenced. And here in Dane County, we're up somewhere between 5 and 10%. We've generated more than 4,000 sequences since the start of the epidemic. And basically all of those have gone through this workflow and have been enabled by the CHTC resources that I described earlier. So what have we learned from this? So I'm going to take you through a couple of stories very quickly to tell you how we've used this data to make insights into how the virus is spreading. And so one of the first questions we had is, how is the virus spreading between Wisconsin? So we were collecting a lot of sequences here in Dane County, and we realized that just down the road, so Madison is connected to Milwaukee by an interstate highway. It's about 80 miles between downtowns of both cities. And what we discovered was there was really no sequencing going on in Milwaukee early in the epidemic. And so we offered our help to the City of Milwaukee Public Health Department to collect sequences from Milwaukee. And we then compared the sequences in the first 2 months of the epidemic between Madison and Milwaukee. And one of the things that we discovered was that the governor's safer at home orders, where people were encouraged to stay at home, actually had an effect on the ability of the virus to spread throughout the state. And we found that the viruses that we discovered in Madison really were Madison-centric. We didn't see those viral sequences in Milwaukee, and vice versa. The sequences that we saw in Milwaukee, we didn't really see in Madison. Furthermore, we discovered that in Madison, the epidemic early on was characterized by multiple introductions of the virus, mainly from European sources. So the viruses that we saw in Madison were most closely related to those that were circulating in Italy, France, Spain, England at the time. And there were multiple different lineages or sort of families of viruses, which indicated that the virus arrived independently in Madison a whole bunch of times. In contrast, in Milwaukee, we didn't see that kind of evidence for multiple introductions from Europe. Instead, what we found was that there was a very small number of introductions of viruses that were virtually identical to those that were circulating in Southeast Asia and China at the time. And so the two epidemics, even though they were separated only by an interstate highway, largely stayed independent. And I think the governor's safer at home order was really important for that. Then as we moved forward a couple of months and the University of Wisconsin reopened in the fall for classes, and students moved into the dorms. Within a few weeks, there were massive outbreaks in UW-Madison dorms, specifically in two high-rise dorms, Whitty and Celery. Here you see a picture of Celery Hall in the middle of September when they were in a 2-week lockdown. And so one of the big questions we had is, if you bring all these students back to Madison in the middle of an epidemic, are the viruses going to infect the students first? And if they do, what kind of risk does that pose to the greater community? And so there was a lot of hesitation about bringing students back. And I was very loud as one of the critics who said that I thought it was very dangerous. And in fact, it seemed like that was being borne out when we had this big outbreak just a couple of weeks into the semester. But interesting, when we sequenced the viruses from this outbreak, we sequenced several hundred viruses and discovered, much to our surprise, that while the viruses that were circulating in these two dorms were virtually identical to one another, which was indicative of spread within the dorms, it turns out that those viruses didn't leave very many descendants in the greater in the greater Dane County community, meaning that these dorms acted as islands, and the viruses didn't really make their way out of these dorms into the broader community. I don't think there was anything more to it than we got really lucky. I don't think this is a generalizable experience. I don't think it's one I would want to relive or repeat and hope for the same outcome again. But the data was very clear that the sequences that were circulating in these dorms basically were extinct in the Dane County area by the beginning of November. So in that sense, we got lucky and dodged a bullet. Just a couple of weeks later, there was a lot of interest about virus outbreaks within intercollegiate athletics. So the University of Wisconsin played Illinois in a football game at Camp Randall on October 23rd. And shortly thereafter, there was public reporting that there had been an infection among the UW team on the UW team, and it turns out that there was actually clusters of cases that were associated with this outbreak, which was really surprising because at the time the football players were receiving what really seemed like the best possible testing strategy. They were getting tested every day, and it was 100% adherence. And so it was really surprising that you would have infections despite this kind of really high frequency testing. And it turns out that what happens is even if you test people every day, you still have an interval of time where someone can test negative in the morning. It can be positive in the afternoon and can potentially infect other people, especially if you don't take other precautions like wearing masks, being outdoors, and so on. And when we sequenced the viruses from the cases that were associated with this outbreak, what we discovered was that they were, in fact, all part of the same transmission chain. So at the time, with so many viruses in the community, we couldn't differentiate ahead of time the possibility that you had 5 different outbreaks within the football team from 5 different sources, or whether they all came from one source in one unlucky event. And almost all, there was one infection that came from a second source, but almost all the infections came from a single transmission event, which means that we have to be really careful when designing these sorts of programs for testing that we don't lull people into a false sense of security, which is, I think, what happened here. And then moving forward, again, we've been working a lot with our hospitals and clinics to try to think about what sort of transmission happens in healthcare settings. And so one thing that's been really reassuring is that since the start of the epidemic, there have been many healthcare workers infected in Madison at our hospital. But when we sequence the viruses from the infected healthcare workers and we compare those to the viruses from the patients who they're treating, in almost all cases, and we've done this about 100 different times now, in almost all cases when healthcare workers are getting infected, they're getting infected in the community. They're not getting infected by caring for their patients, which means that the sort of precautions that are in place and that are being used are actually really effective at preventing a transmission between healthcare providers and their patients. And so I just want to conclude by saying that what we're doing now is we're focusing on a couple of areas of particular interest. We're really interested in sequencing viruses from people who have some form of immune failure. So people who are persistently infected for a long period of time, people who get infected despite being fully vaccinated. This is our friend Laurel giving vaccines here in Dane County. For those of you who aren't in Dane County, I'll take particular pride in noting that we have the highest vaccine rate for any county over 300,000 people anywhere in the United States, which is allowing our county to return to normal much more quickly than other parts of the country. But it also means that people in our community are protecting those who are unable to get vaccinated or for whom the vaccines are not going to be fully protective because of underlying immune compromise or other reasons. So we're doing a lot of work there. We're looking to expand our testing into K through 12 schools where we already are very involved in testing. We want to expand that to sequencing. And then we're also expanding to think about what it's going to look like as case burdens fall and the cases are going to become more rare. So we want to be able to sequence from environmental samples. So we do some sequencing out of wastewater facilities, and we're soon going to be sequencing out of air. So with that, I will stop and ask if there are any questions. Thank you.